In this video, we're going to look at the mechanism of another class of enzymes called the serine proteases. So, we're going to begin by looking at overall what serine proteases are. And they're proteolytic enzymes. And they are common in that they have a mechanism that uses serine. And you usually find serine proteases in digestion, development, blood coagulation, inflammatory, and many other types of reactions. The most commonly studied serine proteases are chymotrypsin, trypsin, and elastase. And what's common between these three proteins is that they have histine and serine that are going to be catalytically essential. However, you also have an aspartic acid that's present and it's buried in a solvent inaccessible pocket. That means solvent cannot touch this asparagine. And when we talk about serine proteases, we often talk about asp, hist, and ser being known as the catalytic triad. So here, asp, hist, ser, all interacting together to do the reaction. These are your catalytic triad. Here we have bovine trypsin, an inhibitor here, so we can see the mechanism in action where we have ser, hisp, and asp all interacting with our inhibitor. And we have something called the specificity pocket to make sure specific serine proteases only work on specific substrates. So let's look at the specificity pocket a little bit more. So different substrates only react with certain catalytic triads. So chyme, or chymotrypsin, only works with bulky aromatic side chains because you have a hydrophobic pocket near the catalytic group. So this hydrophobic pocket will cause the big bulky rings to bind there, and you can chop your bond here to destroy your protein. Trypsin chops after lysis and arges because in its specificity pocket you have an aspartic acid, while elastase will cut small hydrophobic amino acids like al, gly, and val because its pocket has small nonpolar residues to help stabilize this. Now, what's interesting is that if you look at the specificity pockets, there's really not that much difference, especially when it comes to chyme and trypsin. However, if you do a straight mutation of serine 189 for asp 189, you don't actually create trypsin. What you do is you create a crappy non-specific protease. And what this is telling us is that we cannot just look at the binding pocket of an enzyme and expect to be able to understand how it works. There are a lot of interactions that are happening within your enzyme that you may not think is important, but they're kind of long-range interactions. You can have hydrogen bond networks. You can have ionic networks. A lot of these interactions that, when you first look at the enzyme, aren't important, but they are critical for the enzyme to work. And this is really one difficulty when trying to create man-made enzymes. Sure, we can look at the binding pocket and think we know how an enzyme works, but there's probably a lot of interactions that we're not thinking about that are important because these enzymes have been evolving over hundreds of millions of years to be very good at the reaction that they carry out. So it makes sense that many amino acids that aren't directly involved in the catalyst are important for their reaction. Now, looking at serine proteases, chymotrypsin, trypsin, and elastase are incredibly similar how their amino acids are laid out. So they probably arose from gene duplication, and after those genes were duplicated, you have some divergent evolution. That is, you have two copies of the same protein, so if one copy starts to mutate, that's okay because you have an original copy that's still doing the mechanism that you need it to do. But the second the second enzyme can go and do something else. They can cut a new substrate. However, if you look at other serine proteases like subsilin and serine carboxypeptidase, you see that the order of residues, as being shown here, if we look at chyme versus other amino acids, the order that the residues come in are really different. And this is telling us that these enzymes probably had convergent evolution. That is, these proteins aren't related. There wasn't a gene duplication. Rather, nature just figured out separately and independently, oh, these catalytic triads are really good at cutting stuff. So independently, these proteins were formed. That's why you see different orders of amino acids even though they are doing the same mechanism. That's called convergent evolution. Now, let's look at the mechanism of serine protease. So understanding this mechanism will basically allow you to understand all serine proteases, and other enzymes have a similar hydrolytic function. So step 1 is binding the substrate, and after you bind the substrate, serine 195 here will get its hydrogen stolen by the histidine of the catalytic triad. When this happens, the oxygen of serine will act like a nucleophile, and it will bind the carbonyl carbon of your substrate in covalent catalysts. You'll form a tetrahedral intermediate, as shown here. And it's tetrahedral because your carbon now has 4 bonds, and it's in the tetrahedral geometry. So this is our intermediate state. And so this is what our enzyme is trying to bind to, trying to make this lower energy. And so we'll see how we do that later on. But for now, let's continue on with the catalyst. Step 2 is that we're going to take this tetrahedral intermediate and break it down into 2 parts, our acyl enzyme intermediate. This happened by the hydrogen that histidine got for serine. It's going to give it to our nitrogen here and break this nitrogen-carbon bond. So now we have our acetyl enzyme intermediate. This nitrogen group is no longer attached to the enzyme in any way, so it's free to leave. So the amine leaves, and that's replaced with water. So that's step 3. So step 4 is that we have water in our binding pocket, so the histidine, it's going to act like a base, take the hydrogen from the water. That will create a nucleophile on our OH here. The nucleophile oxygen will attack the carbonyl carbon, reforming a tetrahedral intermediate. So this is like step 2, but in reverse. Then step 5 is that it's basically reversal of step 1. Serine will now be taking the hydrogen from histidine instead of donating it. When it takes that hydrogen, the negative oxygen here will reform a double bond to carbon, and this carbon-oxygen bond will break, and now the C-terminal portion of the peptide is free to leave, and your enzyme is regenerated. And note that during this mechanism, it was just a bunch of acid-base reactions by histidine and covalent catalysts by serine. And so asp, which is part of the catalytic triad, didn't look like it was doing anything, but what it's doing is hydrogen bonding to this histidine, which is stabilizing the times when it gets this positive charge and also interacting or modifying its pKa. So although asp isn't actually doing anything via chemistry, you might say, it is forming a hydrogen bond, which is important for histidine to take and release that hydrogen to do its acid-base catalysis. So that is just another example of something not looking like it would be that important in our enzyme, but you mutate it away and this serine protease doesn't work anymore. Now, I said previously that our tetrahedral intermediate is what we want to stabilize. That's what we want to bind to. And to do that, serine proteases will have what's known as the oxyanion hole. So when we have our serine attacking the carbonyl carbene and our histidine taking the hydrogen from serine, we form our tetrahedral intermediate. The way that this tetrahedral intermediate forms is that our negatively charged oxygen is going to go into this oxyanion hole. There, it's going to hydrogen bond with the backbone of a glycine and the backbone of our serine. And you also have this hydrogen bond between the histidine and the amine group of our substrate. And we're forming what are called low barrier hydrogen bonds. That is, the pKa of the donator and the acceptor are nearly the same. So you don't actually have a hydrogen bond. Rather, you're more or less sharing the hydrogen. It's really odd. It's almost like half hydrogen bond, half covalent bond. But this low barrier hydrogen bond, or these low barrier hydrogen bonds, are really why serine proteases are so good. You form this oxyanion hole, low barrier hydrogen bonds. You're really stabilizing this transition state. And remember, the more we stabilize the transition state, the lower that energy barrier, the faster our enzyme works. Right. So these proteins, these enzymes chop up other proteins. So it makes sense that in your body, you don't always want to have these active. Otherwise, it'll start chopping up proteins they're not supposed to chop up. Therefore, these proteolytic enzymes are usually synthesized as large inactive precursors. We call these precursors zymogens. And in fact, if you have acute pancreatitis, what happens is that these digestive enzymes that you're making are actually accidentally turned on, eating your own pancreas. So you don't want that to happen. So our zymogen of trypsin is trypsinogen. And so trypsinogen is inactive, and the way to activate it is that you have to cut between lysine and isoleucine 15 and 16. And the protein that does it is another serine protease, enterpeptidase. This will cut this, and you'll actually cut eventually 6 amino acids from our trypsinogen. What's interesting is that when you make this cut, you're making trypsin, and trypsin can go and work on other trypsinogens as well. So this is autocatalytic. Other trypsins can create trypsins. And blood coagulation, just taking like a wider view of why this is important or where it's used. Blood coagulation uses serine proteases in kind of an autocatalytic form. And you also have a cascade happening. So this is kind of like a big picture view of how a lot of enzymatic processes work inside the body. Is that it's not a simple on-off switch. Rather, you have many layers of control that will turn on a specific process. So let's take a look at this. So we have two ways we can turn on blood clotting, intrinsically and extrinsically. So we're going to start the extrinsic pathway. So you have an injury. When you have an injury, a certain protein is released into your blood called tissue factor, and this is only released if you have broken tissues. This tissue factor will go and interact with what's called factor 7, and you'll create factor 7 tissue factor complex, and this turns on factor 7. So throughout this whole mechanism, wherever you see A, that means activated. All right. So if you have factor 7 activated tissue factor, what this will do is that it will turn on factor 9. So factor 9, and all of these factors are just proteins, so don't get too confused. Try to just follow the logic here. Factor 9 gets turned on to become factor 9 activated. To do this, what you also need is factor 10. So factor 10 will interact with factor 9 to turn into factor 9 activated. In doing that, it will become factor 10 activated. So once we're at factor 10 can take prothrombin and create thrombin. Thrombin is our serine protease. And to do this, you also need factor 5 being factor 5 activated. So once you're at thrombin, what thrombin can do is it can do a lot of things. One, it can go onto the intrinsic pathway and turn on a bunch of factors, which will go and turn on prothrombin. So this is kind of like autocatalytic here. When thrombin gets turned on, it turns on proteins that makes more thrombin, more or less increasing the amount of thrombin. Then, when thrombin is on, it'll take fibrogen and make fibrin. This fibrin is what is creating a cross-like structure that causes blood clotting. It will seal the blood cells in like a net, making sure no more leak from your body. This will also turn on factor 13, making factor 13 activated, which causes this cross-linking to happen. But thrombin turns on itself more and more and more, and you get this big effect. And what actually happens inside a lot of these pathways is that a lot of the upstream events, such as factor 9, factor 11, factor 7, factor 10, are in very low concentrations. So you don't have to make a lot of them. You don't have to spend a lot of energy making all these. But what they do is that their signal is being amplified down the chain. So you have this amplification, till finally you get to the things you need a lot of. And then they are turned on. Thrombin is activated and fibrin is made. So you have these in large supplies, but they're turned on by things that are in low supplies, which is just energetically efficient, and it's efficient with your resources as well. But that's a cascade of a mechanism cascade, and that's how your blood clots, and that's how serine protease is evolved in that. And so when you're going through this, just try to follow the arrows and try to logic out, okay, what's being turned on, when, what's going on, how is this a cascade? And I think just going through it a couple times like that, you should be able to get the process down. Alright, that's it for this video. I will see you in the next ones.